Welcome to the webinar on the use of the handheld 1064 nanometer progeny throughout the pharmaceutical process with an emphasis on its use in a QA/QC environment. Now, we've shown applications for handheld Raman can be used throughout the process control, manufacturing, and other areas in a pharmaceutical environment. But quality control is often a top priority for every pharmaceutical manufacturer, especially if your supply chain changes. So regulatory agencies push for 100% inspection, and manufacturers need to adapt their inspection procedures to put more emphasis on QA/QC in order to test for out-of-spec materials such as incorrect formulations, contaminations, counterfeiting, or mislabeling. Any of these issues can lead to problems throughout the manufacturing process and can result in material waste, reprocessed work, time delays, or even product recalls. So there are several areas through the manufacturing process where fast and accurate material identification and verification, as provided by a portable Raman system, can benefit the users. So specific applications include RMID, which is the first area of common use. It's at the warehouse floor where we have 100% inspection of incoming materials. Next, we can go to QA/QC, which checks for retains and formulated materials and verifies materials used in entire processes. Then we can do secondary process monitoring, where we can verify the final product and also check the incoming materials as a secondary check. And finally, we can look at good application for handheld Raman is the authentication of a finished product. And this can be off-site to detect counterfeit or anywhere to detect adulterated materials. This is especially useful at point of sale, where inspectors can take the instrument off-site to maybe street pharmaceuticals and test for materials. So we're going to illustrate the use of the portable instrumentation throughout the different processes by using a kind of a case study approach. Or we're going to look at the portable system in a number of different areas and just present quick slides on where they can be used. Then we're going to look at a little bit of a larger case study to address the issues of selectivity analysis, specifically for a QA/QC environment. And the first step we're going to look at is monitoring, for example, reaction. So one area of the pharmaceutical process where portable Raman instruments are somewhat underutilized is in process monitoring. So there's a variety of inline instruments that are available to monitor. But for example, the progeny Raman analyzer could also be used as a secondary check for offline or at-line quality processes, where you can take samples at the beginning and during the process and develop an appropriate application on the progeny. So for example, to follow the reaction. So this is a less common technique for portable instruments, but can also serve as a secondary screening technique to give another verification checkpoint. So in this example we have here, we have spectra of two starting materials, epichlorohydrin, which is shown in green in the spectra, and a methoxyphenol, which is shown in yellow. These are starting materials to produce ritonavir. The desired product, which is an intermediate shown in blue, it will then be used in a secondary process to continue to manufacture the ritonavir. But we're just monitoring this small section right now. Okay. Now, if we monitor this, we can clearly see that the spectra are distinct for these materials. So the process can be monitored by following the disappearance of the peaks for the second starting materials, the epichlorohydrin and methoxyphenol, and the increased peak heights and peak presence of the final product, the ritonavir intermediate. Now, as an aside, ritonavir actually has two polymorph forms, and there's another webinar on this showing that these polymorphs can be differentiated also using the progeny. Okay. We can also look at an example for a QA/QC lab other than the process monitoring. Here we see common excipients in the spectra, and we can use the Raman analyzer for verification of these materials. And this is what's done for raw material ID of incoming material inspection in the warehouse. But in this case, we're looking at this part of the QA/QC laboratory process. So QA/QC receives samples from a process, and they have retained samples, and they want to make sure that the materials are what they say they are, and this is the correct materials that have been added. Now, most common excipients are likely to fluoresce with 785 nanometer laser systems. So the advantage of using the 1064 nanometer means that we can actually identify quickly and screen for these materials that were likely to fluoresce before. So here we see the common excipient spectra, and all of these were scanned in pretty well under a minute each one. So it's a quick and dirty way to verify that these are present. So in QA and QC, they would get the sample, they would do a quick check of the materials and verify that they are what they say they are. And as well, they would also might check to see if there are other formulations are correct. For example, here we have some scans where the organic salts have different hydration states. You see a dicalcium phosphate anhydrous, and you see dicalcium phosphate dihydrate. Now, it is important for the physical properties of a formulation that this states are constant and well known. So otherwise you're going to have issues with bioavailability and solubility later on. So one of the other secondary applications QA/QC will be checking for is to differentiate and verify the hydration states. So as an example, if you wanted to differentiate the anhydrous form of calcium phosphate from the dihydrate forms, and you can run them on this on the spectra, and then you can try and build a pass-fail criteria to make sure that you have the correct ones. Okay. So another thing you might be checking here is that the materials that you have scanned are similar to your retained materials. So if you have a new lot, batch, or vendor, you want to make sure that the new material is similar to the material you've put in the library. And one way we can do that is shown in this example, where we can build different analysis techniques to check for the selectivity and specificity of a material. So this would be part of the analytical method validation, and this is covered by publications from USP, FDA, ICH, and other regulatory agencies. So generally the library is built from reference materials, which are usually traceable standards that are well characterized and have certificates of analysis. These tend to be well described by ASTM NIST monographs and also available as a USP or CBER standard. So your initial library is built from these well characterized materials, and then you want to test your retains and your process samples against these. So all of these spectra from the previous slide were put into what's known as a correlation map. And the question becomes then, what should the cutoff threshold be set to clearly differentiate these materials? And our secondary question was, are these materials correct in what they previously were, even though we've changed our vendor. So in this example, we start with a cutoff correlation by default of 0.8. Now, in this case, this should be sufficient to separate out these samples, as the closest challenge materials to the anhydrous dicalcium phosphate, which we're looking for, is the dicalcium phosphate dihydrate, and the correlation for these two is about 0.75. So setting a threshold cutoff of 0.8 is sufficient to separate out these materials. So if the incorrect material is passed and analyzed as what you want, it should fail. So that was good. Then next we're looking at, is there any possible mismatches, or is there any issue because of the different sources of this dicalcium phosphate dihydrate? And that's shown in red, where we compare dicalcium phosphate dihydrate initials, so the retained sample that was in the lab, with the new vendor or batch or lot of dicalcium phosphate dihydrate, and we see that there's a similarity of correlation of 0.99. So there's no problem between these two materials. They're extremely similar to 0.99. We can say that that material is verified as what it says it is, and the other criteria is, and we can differentiate the hydration states from the anhydrous state as well. So this is an example of some of the issues that might be occurring in the QA/QC lab that they'd be testing for. Basically, they're looking to see if there's going to be any, they want the true positives and the true negatives, but they also want to test for false positives and false negatives. Another example that's more recent, in this past year, there's been a general run-on for hand sanitizer, and there's been a lot of adulterated hand sanitizers that have been offered. These can obviously cause health hazards, and they also cause issues because they're adulterated, so they don't work as well as they should. One of the example shown here is for the adulteration of ethanol-based hand sanitizer. So within the past year, and even just recently this past week, the FDA has sent out numerous bulletins about possible adulteration in hand sanitizer. So this not only impacts the efficacy of the material, but it's also a safety hazard because methanol is a poisonous material. So you do not want this in your ethanol-based hand sanitizer. So in this case, we did an examination of methanol contamination in an ethanol-based hand sanitizer. Built an application, and we were able to deploy it on the portable instrument to test samples and this can be used at any scene and verify that they are what they say they are. And in order to do that, to develop the criteria for the pass-fail application, we first looked at the spectra of the correct formulation. And in this slide, you'll see it's in the blue. This is the formulation that we are desired. But when methanol contamination is present, then the spectra is shown in red. Obvious change is the large peak at about 1,000 inverse centimeters. And there's also some minor changes to peak intensity for the smaller peaks. But another change obvious from the spectra is the peak shape at around 1,500 inverse centimeters is also different between the contaminated and the desired product. So obviously, these changes are sufficient to cause a sample to fail. And so we can identify adulterated materials within the finished formulations. So you can use this as a quick screening tool for analysis of any formulation at any on street type of sale, or you can go into a pharmacy and check the materials to make sure that they're not selling adulterated materials. So we've given the example for hand sanitizer, but it works the same way for any kinds of other contaminated or adulterated materials, and for counterfeit materials as well. Okay, so we're going to look in a little bit more detail and expand on an example for the next couple of slides, and to show how a selective library might be built and tested in a QA/QC environment. This application and this library then can be implemented at line of a process prior to adding the materials. In this case, the samples we're testing for are cell culture media. The samples have very similar formulations, but we need a specific type of cell culture media for a fermentation to proceed. Because the wrong type will tend to negatively impact growing conditions and the final fermentation products. So it's very important that the correct cell media be added to a fermentation to make a biopharmaceutical product. Okay. So the cell culture media can be used for the growth of cells in a biopharmaceutical production. Now, Raman spectroscopy is a non-destructive, reliable, efficient, and cost-effective method to analyze cell culture media, and has been recognized by the USP and EP as a viable technique for identification of these cell culture types. So prior to adding the cell culture media to a batch fermentator, the sample can be scanned and verified. Okay. Now, how would an application like that be built? Well, we'll begin in the QA/QC lab. We have standard samples of the common types of the cell culture media that are used in the biopharmaceutical plant. We also include challenge materials that may be potentially incorrect formulations, and see how selective our application is. Now, we're going to look at several examples here, and the most common types of cell culture media are Delvecchio's modified Eagles medium, DMEM, and there's different types with and without glucose, but we're only looking at the ones with glucose. And then there's these F12 types. There's the modified F12 and there's Ham's F12. Now, one of the challenges that we're looking at here is that we need a specific type, but glucose tends to be the majority ingredient for all the types that we're looking at. So this was an additional challenge to differentiate these types of materials. So we're looking at the spectra here. So we begin by taking spectra of the standard samples. And then we ask ourselves, can we differentiate these based just on their spectra? Can we build an viable application to correctly ID the cell culture and then fail the application if the incorrect type of culture is scanned? So look at the spectra. They have many peaks in common. It's actually pretty nice spectra. They're relatively noise-free. There's no fluorescence observed because we're using 1064 nanometer laser. But you can see from the spectra that they all seem to have a lot of peaks in common, and that's probably because the glucose. In general, all of these minimal essential media are composed of glucose as the majority ingredient, salts, which are not Raman active, so that doesn't help us, and then amino acids, vitamins, and others. So we're looking to differentiate these materials based on the smaller components of a formulation. So we began to check the selectivity of these 4 types of materials that we were interested in. So we took the spectra from the previous slide, and we built a correlation map to see how closely correlated all of these spectra were. Now, initially, looking at this data, it was feasible that setting a correlation cutoff of 0.95 might differentiate most of these materials. In this case, if you look at the table, you'll be able to differentiate 3 out of 4 of them just by setting your cutoff of 0 to 0.95. But there is a 0.97 correlation between the two F12 type of materials. Now remember that spectra used for a library are based on optimal conditions. So it's just not sufficient to set the correlation cutoff from 0.95 to 0.97 and assume that all the samples will then be selective. Mostly because we've scanned at ideal conditions. So if we scan at default conditions, which is a quick scan that would be done by a user, the increased noise may actually obscure any differences. So we might not be able to be selective in that case. So what can we do? Well, one option is we can build a custom cascade application to further differentiate the two types of spectra that are correlated at 0.97. And we can do this by looking at those two materials. So they're both F12 cell media, and they both contain glucose. Now the only difference between the two of them is actually that modified F12, shown in orange here, actually contains arginine and sodium pyruvate. So can we examine the spectra and try and find some difference in the spectra between the two of them? I would do this looking for the presence of this arginine and sodium pyruvate. And we find that there might be peaks due to the primary and secondary amines that are present in one that are not present in the other. So if we look at the two extra peaks in the modified F12 that could be due to an arginine, they are present. There's an C to N stretch at about 967 inverse centimeters. And then there's the other amine stretch that characteristic bands for are at 1412 inverse centimeters. So it is possible that we can use these two peaks to differentiate the two closely related cell media. So we could build a secondary application to separating out these two types, and then based on a spectral overlay with the library materials, we can check for the possible presence of these two peaks. That may not be a perfect solution, but because of the close correlation, a cascade model might be the best choice for this one. It can certainly be written into an SOP that any QA/QC lab can use. Remember, we were not looking at pure screening here, but rather we're in an analytical laboratory looking for a quick check of our materials. So we're basically looking at an SOP that involves a two-pronged approach for cell media selectivity. We built the library for the common types. We verify them quickly in under a minute to verify. And then we can overlay the spectra if the F12 materials show up and check for certain peaks to further narrow down which material it is. Again, overlaying the material would take maybe another minute or two. So you're still looking at under 5 minutes to verify that your material is what you think it is, and it is, and is it the correct material that's going into the fermentation broth. So this is still pretty well a rapid test in a QA/QC environment using our portable progeny instrument. So to summarize, hopefully from this presentation, we have shown how a portable instrument such as the progeny can be used as an integral part of the pharmaceutical process, beginning with RMID for incoming inspection and integrating into the larger parts of the pharmaceutical process. We can verify materials at process lines. We can monitor a reaction process. We can monitor the formulation and check and verify that it is what it is. And we can also implement this as part of the QA/QC analytical laboratory. And basically, because it is portable instrumentation, the portable nature allows inspectors to take it out to offsite and to street samples to check for adulterants and counterfeits. And these are just some of the potential uses for the progeny. And I would like to thank you for your attention. And if you have any questions or need more information, please feel free to contact me at my email address. And thank you for your attention.